Hello, and welcome back. So this is our signaling pathways continued. I want to kind of talk about the four types of receptors in a little bit more detail. So let's take a look at this video first.
Hi there, and thanks for joining us as we get a quick look at the four types of cellular receptors. Three of the four types of cellular receptors are on the cell surface. They're called transmembrane proteins, meaning that they span the whole distance of the phospholipid bilayer from the outside of the cell to the inside of the cell. So let's take a quick look at each one of those cellular receptors. The G protein receptors are membrane bound receptors. They're also called 7-transmembrane receptors, and they're the largest and most diverse group of membrane receptors. Binding of the ligand or of a medication mimicking the ligand results in the generation of a second messenger, which is involved in cellular signaling. Another membrane-bound receptor is the enzyme-linked receptor, also called tyrosine kinase receptors. The insulin receptor is an example of that type of receptor. Binding of the ligand or a drug mimicking the ligand, simplistically speaking, turns an inactive molecule in the cell into a molecule that starts signaling the cell to undergo a series of actions. The other receptor that is a membrane-bound receptor is a ligand-gated ion channel. And when the natural ligand binds to that receptor, the protein undergoes a conformational change again, but that change simply opens up an ion channel, and that allows positively or negatively charged particles through the cell surface. Those are the three types of membrane-bound receptors. The ligands that affect those receptors can access the receptor from the outside of the cell, meaning that the ligand or drug binding to the receptor can be water soluble or lipid soluble. However, the ligands for the intracellular receptor need to be lipid or fat soluble in order to get through the phospholipid bilayer. When those receptors bind to the ligand, the entire receptor ligand complex goes into the nucleus of the cell, binds to the DNA, and affects the transcription and translation of certain portions of the DNA. Those were the four types of cellular receptors, and we'll find out more about how our medications affect those cellular receptors in the next few lessons. Thanks for joining in, and we'll see you there.
Okay, I know that guy's voice was a little weird, and we're not really talking about drugs, but the information that was given in the video and the animations are just perfect. So let's talk about the G protein. So before I even put any notes up, let's just take a look at what's happening in the diagram. You can see here that the G protein is inactive because there is no, I say ligand, that's just how I pronounce it, because there is no ligand. So here we have in the second part, part B, you can see that the signal molecule binds to the receptor protein. In doing so, it activates the G protein, which then pops on over and activates an enzyme. Then through several transduction steps, we have finally a cellular response. Then when the ligand is detached or, is transmitted the message, then it returns to its inactive form. So that's all that's happening with the G protein. The signal activates the G protein, which then goes over and activates an enzyme, causing some sort of cellular response. So what kind of ligands or ligands can activate G proteins? They can be neurotransmitters, hormones. That would be a protein-based hormone, however, because it would not be able It binds to a surface receptor rather than passing through the plasma membrane. The transduction, or the processing of that signal that leads to a cellular response, includes the signal activates the protein, which then activates an enzyme, generating a cellular response. So that's G protein. All right, next we have the ion channel receptor. So again, before I even show notes, let's just look and see what's happening in the diagram. So you can see in the first panel, the gated ion channel receptor is, it's closed. And then once the signal molecule binds, it causes that conformational or shape change in the protein, causing the channel to open. And now ions can pass through the cell membrane. The presence of those ions will trigger that domino effect or that cascade effect, which will then lead to a cellular response. Once the message is no longer bound to the receptor, the channel closes, no more ion flow. So what kind of ligands open ion channel receptors? Neurotransmitters. The transduction, or the process of the message once it's received a cellular response, the signal opens the channel, ions flow through. That increased concentration will lead to some sort of cellular response. The tyrosine kinase receptor, I say kinase, I'm from East Tennessee. So the tyrosine kinase receptor, you can see that it's actually made up of two receptor proteins. And so while they're hanging out waiting for their signals, you can see that they are inactive, and they have some distance between them. Then once the signal molecules attach, in this case it takes two, one binding to each of the proteins, they will kind of join together. Then they phosphorylate or add a phosphate group to these relay proteins, and then the relay proteins again cascade effect, activate other molecules until we finally generate some sort of cellular response. So what type of ligands activate tyrosine kinase receptors? Growth factors. Transduction. The signals will bind to two receptors, triggering several relays and responses all at once. So anytime we talk about transduction and cellular responses, just keep that mental image of the dominoes in your head. It's a domino effect. One molecule activates another and another and another until at the very end we have some sort of cellular response. All right, finally we have the intracellular receptor. So again, let's look and see what's happening in the diagram before we put up some notes. So in this case, we have a lipid-based hormone, testosterone, which is one of the sex hormones. Estrogen is the one for females. And so since it is also nonpolar and it's lipid-based, it can slip right through the plasma membrane. It's going to bind to an intracellular receptor somewhere in the cytoplasm. Once those two things bind, they can then pass through the pores in the nuclear envelope, go into the nucleus, and then they will switch on genes, which will then generate a protein. Now, we've got a whole unit on how proteins are made, but for right now, let's just kind of be like, okay, this is going to switch on a gene, we're going to generate a protein, and that's the cellular response. So what kind of ligands are bind to intracellular receptors? Steroid hormones. Again, estrogen and testosterone. These are lipid-based hormones. Transduction, the signal passes through the membrane, triggers a change in the cell, and this turns genes on or off. Now, I have in a built-in review activity. Obviously, working at home, you don't have a team to work with, but what you could do is just gather up some scrap papers and first just write down two receptor types, and then any two transduction summaries, and then write down the two other receptor types and the other two summaries. And then if you'll just turn the papers face down, mix them up real well, turn them back up, turn them back face up, and just work on matching them up. And match them as many times as you need to until you can do it just without even looking. All right. Thank you for your time and attention. Until next time.